Okay, students, so I welcome you back to another important lecture. And in today's lecture, we would see the second component that we have in case of a transport system in the humans. And that component that we need to be discussing today is the blood, because blood is something very important, as this blood is actually taking the nutrients from one part of the body to another part of the body and also distributing the waste materials to the kidneys, and this is actually the blood which would be flowing within the blood vessel. So after we are done with the heart, in the previous classes, so now we would look on to what this blood actually is, what components or what would be the materials that we would have in this blood. Now, for blood, what you have to keep in your mind is that this is a connective tissue. Why we call this as a connective tissue? Because this is bringing a connectivity between the multiple organs of the body. If my pancreas was producing the insulin and it has to be carried to the liver and the muscles, so the pancreas itself will not walk to these organs, but rather it would give the insulin to the blood, and that blood would be carrying it to the target organ of that hormone. So this blood is important in terms of that it is bringing a connectivity between the multiple organs that we have in the blood. Now, if I were to tell you that what we have got in the blood, so the major component would be formed by the blood plasma in the blood, and then we have got major of these blood cells with us, and then the small proportion would be also formed by these platelets that we have. So in the blood, like overall, we have this blood plasma, then we have these blood cells, and then we have these platelets. Among the blood cells, we further have these red blood cells and the white blood cells. The red blood cells, technically, they are referred to as erythrocytes, and the white blood cells, the other name of them is the leukocytes. And in the white blood cells or the leukocytes, we further have got two subcategories or the two subdivisions, and they include the phagocytes that we have with us, and then we have these lymphocytes coming with us. Now, if I were to show you with the help of this diagram, so if I say that from some volunteer, I have taken some 100 cm cube of the blood from out of its body, then the 55% or 55 cm cube of that person would be this plasma, and the remaining 45% or 45 cm cube would have the red blood cells, it would have the white blood cells, and then there would be the platelets in it. So this means that this plasma is actually the yellowish liquid part of the blood, and in this yellowish liquid part of the blood, we have got multiple of the materials dissolved in it, and this plus plasma is also suspending the red blood cells and the white blood cells. And we will see in a while that how does the morphology or the structure of these blood cells they actually look like in a while. So if I were to tell you that how this blood plasma is important and what composition this blood plasma has in itself, so overall this makes 55 to 60% of the blood. And if we have to describe it, so this is yellowish liquid part of the blood. Among the various components that it has in itself, so we would have this water, and blood plasma is itself 60%, but the water in the 60% is 90%. We would have the nutrients dissolved in it, like the amino acids and the glucose and the vitamins. They would be dissolved here again in the blood plasma. Among the waste materials that this blood plasma would have in itself, it would have a lot of things like we will have this carbon dioxide waste material, we would have this urea coming, the uric acid being produced, and the creatinine that is produced in the muscle. So again, they would be available within this blood plasma. And the blood plasma also has some hormones dissolved in it, like the insulin and the glucagon and the antidiuretic hormone that has to be carried from the organ that produce it to an organ where they have to act, which is regarded as a target organ. So again, this blood plasma would be used. Again, in order to fight against the diseases, we have to be producing the antibodies by the lymphocytes. So this again would be carried by the blood plasma. Apart from that, the minerals and the vitamins and the blood clotting proteins that have the prothrombin and the fibrinogen proteins in them. So again, they would be available within the blood plasma. So this blood plasma makes the major part of our blood. Now, moving onward, you see that out of 100%, 55 to 60% is the blood plasma, and the remaining are the blood cells with us. And if I were to show you this diagram, so you see here that the red blood cell and these white blood cells that I have with me, so they are being suspended in this blood plasma. So they appear as a suspensions in the blood plasma, and the major blood cells that we have got in this blood. So we will have these red blood cells, and the other name of these red blood cells is erythrocytes. Then we have got these white blood cells, and the other name of the white blood cells is leukocytes. Now, this is not required, but yet you must know for your higher classes that these blood cells generally they are categorized into being called as a lymphocytes and to be called as a phagocytes. And in the phagocytes that eat up the pathogen, further we have the neutrophil and the macrophages. So these names are actually not required, but yet you must know them. But these lymphocytes and the phagocytes name should be there in your mind. Now, if I were to look on their structures at how come the red blood cells and the phagocytes they actually look like, so if you just see on this diagram, so here we have this red blood cell, and this red blood cell at the center they are pinched, so they are the biconcave discs. And if I were to tell you about the white blood cells that we have coming in our body, so, among the white blood cells that act as the phagocytes that eat up the pathogens, so you see that the usual one among the phagocytes is this neutrophil. And this neutrophil has got multiple lobed nucleus in it. And at the same time, we have got these intracytoplasmic granules in them. So they are the phagocytes that are supposed to be eating up the pathogens. And if we talk about the lymphocytes that we have here, so these lymphocytes at the center, they have this nucleus, and if you were to see this nucleus, so this is covering up most of the space of the cytoplasm. And these lymphocytes are important because they remember that what pathogen or the disease-causing organisms has attacked us, and at the same time, these lymphocytes they would be producing specialized proteins, and they are referred to as antibodies that would overcome the pathogens whatsoever would enter into our body. Now, here is another diagram, and this can be given in your exam where you are required to be identifying that what blood cell we have with us. Now, you have to be identifying the white blood cells if they would have the nucleus in them. Like if I were to look on this white blood cell that have the nucleus and this does not have the lobes in it, so I would say that this is a lymphocyte. And if I would have this cell coming with me, and it has a nucleus in them at the center, but this is a multiple lobed nucleus. Like if I were to be redoing it, so this is how the multiple lobe nucleus looks like. So this would be the lymphocyte with me. And the red blood cells, they are very easy to identify. Like if you were to see here, so they don't have any of the nucleus. So this is red blood cell. And in the same slide that they have given us, so the small fragments that we have got, they are not the actual cells, but the kind of the fragments, and they would be referred to as the platelets, we say. Now, another frequent thing that comes in your exam is that, okay, if we have the red blood cells, and if they are carrying the oxygen from the lungs to the tissues of the body, so how their structure would be related to performing this function of delivering the oxygen gas to the tissues of our body. Now, the first point is that these red blood cells they are the biconcave discs, and by the biconcave discs, what we mean is that these red blood cells that we have with us, so they are pinched at center. So when they become pinched at center, their surface area to the volume ratio would be increased, and they would become very efficient at not only capturing the oxygen gas from the alveoli of the lung, but also at delivering that oxygen gas to the tissues of my body. The other thing is that these red blood cells they have a protein in it, and that protein is the hemoglobin protein. And that hemoglobin protein basically has got in itself some you could say non-protein part, and that non-protein part is iron with us. And the role of this iron would be that whenever these red blood cells they would be there in the blood capillaries of the alveoli, so they would go further and then they would bind with this oxygen gas that we are inhaling. Now, the scenario is that the overall function of the red blood cell is dependent on this hemoglobin protein. Then we need to pack more of this hemoglobin protein in these red blood cells. So, for that purpose, what the red blood cells they do that during their development from the red blood cells, the body would remove the mitochondria and the nucleus and the ribosome, so it would create more space so that this hemoglobin protein should be abundantly packed within them. And another scenario is that these red blood cells they have an ability that when they pass through the blood capillaries, so they squeeze through them. So when they would squeeze through them, then as a result of that, their contact with the surrounding tissues would increase. And that is why they would now deliver more of the oxygen gas, or they would deliver efficient amount of the oxygen gas, or the sufficient amount of the oxygen gas to the surrounding tissues that we have with us. Now, moving further, here we would look into the detail that how come the type of the white blood cell that we were referring to this as phagocyte and the lymphocyte what they are doing. For the phagocyte, what you have to understand is that they would eat up, they would literally eat up or they would ingest the pathogens. And the lymphocyte that we have, so they would produce antibodies and they would also produce the memory cells that would remember the pathogen. Now we were done with the diagram. But you see that for the phagocytes that we have further, we have two subdivisions. We have the neutrophils in them. Then we have got these macrophages. The neutrophils, which is one of the phagocyte, that this has multiple lobe nucleus. And if I were to talk about the macrophage, so they would have a nucleus, and this nucleus would be oval-shaped nucleus. But no matter if we have the neutrophil or a macrophage, which are the subdivisions of the phagocytes, so what they have to do is they have to eat up the pathogen. Now, the scenario is that my body would always be at a risk of catching the pathogen. And by the pathogens, what we mean is that they could be the bacteria, they could be the funguses, they could be some protococs, they could be the viruses, and what they do is that they would cause a disease in the human body. The usual thing with these pathogens is that help the white blood cells to be recognizing them, that whether this is something foreign or whether this is a normal cell of the body. So on the surface of these pathogens, we have these proteins, and these proteins they are entitled as antigen. And thanks partly to the presence of these antigens, that would help our white blood cells to carry some reaction against them and not against the normal cells that we have in the body. Now, quickly we would see that okay, we have these phagocytes among the white blood cells. So how they would eliminate or get rid of the pathogen which is entering into my body. So they do so by the help of the process, and that process is regarded as a phagocytosis. So if I were to tell you the definition of this phagocytosis, so this is a process by which the phagocytes that we have, so they would eat up or they would ingest the pathogens that are coming into my body. But at the same time, I have to be ensured is that these phagocytes, they should not themselves be killed, but they should only be killing the pathogen. So now the first point would be that any of the pathogen that has entered into my body, they would initially make the attachment with the cell membrane of these phagocytes. Now, after they have got attached with the cell membrane of the phagocytes, what these phagocytes would do, that they would enfold their membrane inwards. And when they would enfold their membrane inwards, so they would pack this phagocyte in a vacuole. And now you see that we have actually isolated the pathogen from the cytoplasm of the phagocyte. Now we need to be digesting them off. We are required to be killing them off. So in these phagocytes, we again have got some vacuoles, and these vacuoles they have some hydrolytic enzymes in them. So these hydrolytic enzymes containing vacuole, they would fuse with the vesicles that have the pathogen in it. And when the enzyme they would be released out of these, so that would clearly be killing up or digesting this pathogen off. So in this way, the body would be saved from the any of the harm that these pathogens could be causing. So if I were to like simply say these points, so you would say that in the phagocytosis, first is the attachment of the pathogen with the cell membrane of the phagocyte. The second point is that the cell membrane would enfold, and this would enclose a pathogen in a sac. And then there would be binding of the vacuoles here that have the hydrolytic enzymes in them so that they can be digesting the pathogen off. And when the digestive enzymes would be released out of these vacuoles that we have, so ultimately the pathogen would be killed and our body would be saved from any likely harm that this pathogen can have on my body. Now, lastly, among the lymphocytes, we have got these further two subtypes of the lymphocytes. One would be the plasma cells, and then there would be these memory cells. The plasma cell is not the word that you have to know, that you have to remember, but yet you must know for your future classes that these plasma cells they are a further subtype of the lymphocyte. And what they do is that they would release antibodies out of them. And when these antibodies would be released out of these plasma cells, so they would help to eliminate the pathogen. The other situation with these lymphocyte cells that some of them they would be acting as a memory cells and they would keep a memory of the pathogen that has attacked my body for the first time so that later in life when the same pathogen would enter again, So these memory cells, they could initiate an immune response or a reaction against them immediately. And by that, the tendency to which my body would be getting harms, it would be reduced. So I hope you would be clear with the blood, with the structure and the function of the red blood cells and the white blood cells, and also their morphology.